Welcome to the third screencast on the diet and food production unit of F212. This screencast focuses on using microorganisms to make food for human consumption, and then latterly it looks at avoiding food spoilage. So dealing with microorganisms and food for human consumption, I've split this into two parts. First, we're going to look at traditional methods of using microorganisms for food production, so things like yeast, which is a single-celled fungus, which is used to produce in the baking industry to make dough rise, and also used in the alcoholic beverages industry to make ethanol and carbon dioxide. Okay, and we'll talk about some of the advantages of using yeast in that way, as well as we can use bacteria in the production of cheese and yogurts. Okay, and we'll look at a little bit of that too, and look at the advantages and disadvantages of using microorganisms in that sort of way. And then my second half of this slide will look at more up-to-date or more recent biotechnological processes which uses fungus called to produce what we call mycoprotein, okay, which can be used as a meat substitute. Okay, and we'll look at that, and we'll look at the advantages and disadvantages of mycoprotein production. Okay, so first of all, dealing with the traditional use of microorganisms in cheese production and alcoholic beverage production, etc., we'll just have a look at this short video. And secondly, as I explained, the use of fungus, which can be grown on agricultural waste products to produce mycoprotein, a more recent biotechnological process, and some of the advantages and disadvantages of that. Like any biotechnological process, using microorganisms is a fast rate of production. Okay, we've got no animal welfare issues here in terms of producing this product. Okay, and therefore the protein is suitable for vegetarians. And the other nutritional advantage is that the protein that's produced has got no animal fats in it. Okay, and as we know, saturated animal fats, what they do is that they can increase LDLs, which transport cholesterol to arteries to cause atherosclerosis, okay, and also is free of cholesterol as well, dietary cholesterol, which needs to be controlled for some people. Okay, so those are the advantages. Disadvantages: it can be quite difficult to isolate the protein from the agricultural waste products on which they grow, and therefore the purification is expensive. Okay, and there is also a danger, as with any microbiological process, that you have pathogenic microbes could also be growing on your agricultural waste. Okay, and this last one is palatability. Okay, the material, this mycoprotein, doesn't taste and doesn't have as nice a texture as traditional meat products. Okay, so if you've ever tried the vegetarian bolognese sauce on offer at lunch, which is made of corn, which is a trade name for a mycoprotein, the texture isn't the same as what you would be used to in the normal mince you'd get in normal bolognese sauce. Okay, so that's what palatability means. Okay, texture and taste isn't as nice as traditional meat products. Okay, so that looks at traditional use of microorganisms to produce beer and bread and cheese and yogurt and things, and the advantages and disadvantages of that. And it also looks at more recent biotechnological processes to produce mycoprotein from agricultural waste as food for humans. Okay, moving on to the next slide. This slide talks about how to prevent food spoilage. Okay, food will spoil, and what I mean by spoil is it will start to decompose by decomposing microorganisms such as bacteria and fungi if it's left lying around. Okay, we call that food spoilage. Okay, and this is a huge problem. Okay, potentially a lot of the food that we produce in agriculture will go off or spoil before it actually has a chance to be consumed by humans. Okay, a massive problem and a massive amount of waste of food because of food spoilage. Okay, so throughout time we have looked at ways of reducing food spoilage. Okay, and the basic premises of reducing food spoilage is to reduce the conditions, or to remove the conditions necessary for the growth of the microorganisms. Okay, so microorganisms basically need water to grow, they need warmth, they obviously need a source of food, and they need oxygen. Okay, and if we can remove those, the microorganisms won't grow, and so that we can reduce food spoilage. Okay, so those form the basis of strategies that we use to try and reduce food spoilage. Okay, and there's various ones we can do. Look at salting. Okay, so what salting does is it reduces the water potential around our decomposing bacteria and fungi, and so water will be withdrawn from the bacteria and fungi, and therefore they don't have that water that they need to be able to multiply. Okay, so that's what salting does. Sugar conserves sort of things like jams and so on, which use very, very high concentrations of sugar. Same strategy, that reduces water potential, withdraws the water from the bacteria and the fungi, okay, which renders them unable to reproduce. Okay, so that's salting and adding sugar. Pickling. Pickling is putting things in high acid, so vinegar. We place things in vinegar and that reduces the pH, creates a pH which kills bacteria and fungi. Okay, so that's the whole strategy behind pickling. Okay, freezing. Okay, if we freeze our substances, so freezing that actually converts all our liquid into a solid ice. Okay, so obviously bacteria can't multiply at all in an environment where everything is in a solid form. Okay, so that stops bacteria and fungi, and we can preserve food for very, very long periods of time by freezing it. Okay, heat treatment. Okay, so heat treatment, what we do is we kill bacteria by heating it up. Okay, so we can pasteurization. If you go and look at milk, it all says it's pasteurized. We heat that up to 72 degrees centigrade and then cool it down to 40 degrees centigrade. And what this does is it kills a lot of the bacteria in the milk. It doesn't kill all the bacterial spores though, therefore the milk will eventually spoil, but it does kill a lot of the the bacteria in the milk, therefore prolonging its shelf life. Okay. There's another type of heat treatment called ultra heat treatment, which is heating it ultra under pressure, and heat treatment, ultra heat treatment. Okay, heating under pressure, and that kills bacteria and spores. Okay, so the canning industry works on UHT. So what we do is that we heat our food or soup or whatever it is up to very, very high temperatures under pressure in a big pressure cooker, about 120 odd degrees, which kills all bacteria and all bacterial spores. And then we seal the food in a container into which oxygen and microorganisms can't get into. Okay, so we have a completely aseptic environment inside our can. Okay, so that there will be no food spoilage in that can until we open the can. Okay, and then lastly, irradiation. Okay, we can use certain types of radiation can kill microorganisms by disrupting their DNA. Okay, so those are all outlining how various methods can be used. Okay, and then I've gone through them again here. I've added some other ones here. Okay, refrigeration, that cools, it doesn't freeze things, but it cools, so it slows the rate of bacterial multiplication, so food lasts longer. It doesn't stop it going off, but it makes it last a lot longer. So your milk, for example, that you put in the fridge can last 4 or 5 days, whereas if you leave it out on the kitchen bench, it only lasts 1 or 2 days. Okay, freezing, we've talked about freezing. Okay, stops the growth of microbes completely. Okay. Salting, okay, salt forces water to leave the cells, including those of your microbes, and therefore the microbes die. Okay, pickling, vinegar is an acid, creates an acidic environment which kills bacteria. Okay, and then canning, food is boiled, UHT, ultra heat treatment, kills microbes, and then sealed. Okay, and the cans are pressurized to destroy any endospores. Okay, so it's ultra heat treatment that we put our cans into a great big pressure cooker, which creates a high temperature, which is capable of killing bacterial spores. Okay, so that summarizes all of that. Again, I've got a little video here to finish this topic off with, which goes through some of these processes.
Microbes are responsible for creating some of our most enjoyable food products, such as cheese, bread, yogurt, and of course, beer. But other microbes, such as Salmonella, Listeria, or E. coli 0157, can do us a great deal of harm. In the UK alone, there are more than 10 million cases of gastroenteritis each year, a concoction of symptoms such as nausea, vomiting, diarrhea, and stomach pain caused by eating and drinking food contaminated by microbes. Since ancient times, we have done our utmost to keep them out.
If you leave fresh food lying around, one of the first things that happens is that microbes land on it, and they start to deteriorate the food over time. And one of the worst things they can do is start to produce toxins that can be poisonous to anyone eating that food. And the ancients discovered a very, very long time ago that one way to stop this is to try and preserve food.
They used a variety of methods, such as salting, drying, icing, pickling, smoking, and fermentation for beer, cheese, and bread, all of which we still use today.
You can deny microbes water by turning the food very sugary, for example, making jam. Or we can put salt on our food, and that's why people used to salt meat in the days before refrigerators to stop microbes from growing. One of the characteristics of microbes is that they're very sensitive to temperature. Many of them don't like cold temperatures, many of them don't like hot temperatures. And so we can preserve food by changing its temperature. And one way in which we do this is to put food in a refrigerator. We also boil food. That's a very common way to preserve it. And the way in which that's working is to kill microbes that can't really grow at high temperatures.
Boiling doesn't kill all the microbes, just most of them. Some bacteria are actually resistant to the temperature of boiling water, which is 100 degrees Celsius. To get rid of them, you need to raise the temperature to about 121 degrees, but you can only do this by heating water under pressure. A common method of food preservation using this technique is canning, which was developed by the French confectioner Nicolas Appert. In the 1790s, a pair began experimenting with ways to preserve foodstuffs, succeeding with soups, vegetables, juices, dairy products, jellies, jams and syrups. He placed the food in glass jars, sealed them with cork and sealing wax, and placed them in boiling water. And then there's alcohol and acid.
Many microbes can't survive alcohol, whereas yeast can, which makes it an effective form of preservation. Acid conditions, such as vinegar or pickling, are also inhospitable environments for microbes.
Pickling to keep out microbes is responsible for some of our most famous brands. In 1922, in Burton, Staffordshire, Cross & Blackwell produced the very first jar of Branston pickle. 28 million jars of it are now sold every year, still keeping out the microbes as it did 90 years ago.
One thing you have to remember is that if you refrigerate food and the microbes on it, you only slow down the growth of the microbes. You don't actually kill them.
So, when you get back from holiday, all might not quite be as you left it.